All right, everybody. I think it's time for us to get underway. Welcome back. Good to see so many of you survived that first test. That's what I thought about it. Let me tell you. We'll start today just by talking a little bit about that first test. What you're seeing on the screen right now is just a distribution of the grades for the students, and that was like 3/4 of the class who looked at the first exam prior to when it is that we closed the door on that extra credit deal, right? So if you remember how much I had been harping, look at the old tests, that sort of stuff, use Pilot, look at the old tests. If you're in the population of students who did that, I think you did pretty well. The distribution there is what, about 72% was the average. There were 4 people who got perfect scores, pretty much happy day there. An awful lot of people in the 90 to 100 range, which would certainly correspond to an A. A lot of people in the 80 to 90 range. And there's some people down over here that we need to figure out something to do a little bit better for. And this is in contrast then to those who did not look at the first exam. The average is 10% less. There weren't any perfect scores, and I think, look, I'll toggle back and forth. Whoa, see how what's going on there? If you didn't follow my advice and you didn't look at the first test, there's a skewing significantly to the left-hand side of that distribution. The overall average for the class ends up being just a little bit below 70%. And I'll tell you, based on how things have gone with previous times I've taught the class, 70%'s meaning we're doing all right. That's kind of consistent with what I've seen in previous years. So I think on average, things are looking pretty good. I'm not picking up on something that's shouting out to me that there's a serious disconnect between me and you at this point. So again, on average, I think things are looking pretty good. And maybe the one point to really take away from that toggling back and forth between this distribution and the other distribution is that if you're not happy with how you did on the first test, the single best thing that I can suggest to you, that I know that I can suggest to you, is start getting ready for the second test and start looking at some of those old second exams that are available on the Pilot page of the course. I think that perhaps some of the people who might not be that happy with how they did on the first test, who did look at the first test before the deadline for that 1 point of extra credit, I think maybe they didn't look at those old tests in the right way. And you know who you are, right? So maybe what you want to do is stop and think like, okay, I got the 1 point of extra credit for opening the file, but now maybe I should sit down and spend some time looking at what's in those files or something like that, and then that might make the difference. I'll tell you this, that if I had to assign grades for the course right now, based on this distribution, I don't see a screaming, crying need for much in the way of a curve. So the syllabus spells out some cutoff points for grades. And again, if I had to assign grades right now, I'd probably stick pretty close to what's spelled out in the syllabus. 90 to 100's an A, 80 to 90's a B, that sort of thing. But I wouldn't base grades just on one test. Obviously, we've got another midterm coming up in another month, later this month, right? Or maybe at the beginning of November. And then there's a final exam. So there's still a whole bunch of points yet to go. So again, I wouldn't curve any specific test, but what I will do at the end of the semester is I'll look at your whole body of work, and then I'll look at the whole class's overall scores, and we'll see if some adjusting needs to be done then. But I'm telling you, don't come away from this lecture thinking that, oh, I got a 60, but it's going to work out all right, because I'm not going to be curving anywhere near down that low to pull you up to a C or anything like that. So that's where things are right now. In terms of the test itself, I get all sorts of statistics back when we have the computer grade them like that. And there were a couple of questions that I was surprised, I suppose, that some people had trouble with. Question number 7. for instance. So I've got a copy of the test here with me. I presume you guys still have yours somewhere. If you don't, you can find it on the pilot page for the course. But question 7, I'm just going to talk about 4 or 5 questions, 5-ish questions, just to make sure that we're all on the same page, because I was a little surprised at the low number of students, the small number of students who picked the right answer. So question 7 was about elements having their own set of properties. They can be combined in specific ratios. Which of the following statements is true of all naturally occurring elements? What we were looking there for the correct answer is that atoms with the same number of protons behave the same chemically. Okay, so that's the right answer. 45% of the class agreed with me that that was the right answer. I'm not sure what was happening for the other 55%, but I hope that now you'll agree with me that it's the number of protons that ultimately determines what atom you're talking about. So carbon-12 has 6 protons and 6 neutrons and 6 electrons, right? Carbon-13 has 6 protons and 7 neutrons, 6 electrons. They behave the same chemically, and it's because, in some level, they have the same number of electrons, but what determines the number of electrons is the number of protons. So if you were talking about something with 8 protons instead of 6 protons, you'd be talking about a different element. It's the number of protons that ultimately is winning the day in those sorts of things. And then, skipping along to question 20. This is one of the ones where I had to hand draw in a structure. 32.7% of the class got the right answer for that one, so I think this may be the lowest of all. There are only 4 choices, and so if you were just blind guessing, you had a 25% chance of getting it right. 32.7% isn't much above blind guessing. I'll confess to you guys that as I was putting together the test and as I was generating an answer key for the test, that I looked at question 20 and I thought, that's pretty hard, right? A was the right answer there. I showed you a phosphate group, but it wasn't quite a phosphate group because one of the oxygens was missing. So I'll confess, that's a kind of, below the belt kind of question. It's fairly specific. The other 3 structures were pretty unequivocally things straight out of what we had talked about in lecture. The one that was wrong, the phosphate group, I felt guilty about that one. But nonetheless, a lot of people did get it right, and we had some perfect scores, so I don't feel that guilty about it. And at the end of the semester, maybe that'll be one of the things that causes me to be motivated to have some sort of curve, the pangs of guilt that I have for question 20. For question 25, the question was, which of the following functional groups is most likely to impart a hydrophobic patch to organic compounds in which it is found? The right answer, by my reckoning, is C. Only 37% of the class agreed with me about that. And I don't know what happened there. I thought that was a pretty easy question. Can anybody tell me? I mean, your other choices were phosphate. That's not hydrophobic, that's hydrophilic. Carboxyl, pretty hydrophilic. Sulfhydryl, at least polar, so slightly hydrophilic. And alcohol, polar, so at least a little bit hydrophilic. Methyl's carbon with a couple hydrogens on it. So I guess I don't need to understand why you chose something else, but just so long as you're all with me now thinking that, oh, okay, carbon to hydrogen bonds, hydrophobic, nonpolar, that would be a hydrophobic patch. Right? So again, I just wanted to make sure we're all pretty consistent about that. And then, let's see, question 30. Something like 40% of the class got it right. The right answer was catalytic activity. What I did there was draw out for you the structure of a protein. I think it was a dipeptide. It might have been a tripeptide. You can see I don't have it on the version that I've got here. But nonetheless, that's what a protein looks like. And 31, I asked which of these following guard groups looks like they would be hydrophobic, is pretty much what the question comes down to. And it was the one that had a methyl group associated with it. It's D. So maybe there's a recurring theme here of sorts in that I didn't emphasize enough to you guys that a methyl group was hydrophobic. Nonpolar covalent bonds will be afraid of water, won't want to interact with water. Let's move on from that point. Okay? So, any questions for me about this test? If you want to look at your Scantron, I got no problem with that. Come catch me during my office hours. I've got the big stack of them sitting there waiting for people to look at. They are in no particular order, so if you want to see yours, what you got to do is pretty much set aside 5-10 minutes for the idea of looking through the stack to find yours, and then it's another minute or two to grade it by hand. I'll tell you, based on past experience, it seems that every exam, about 2-3% of the class wants to take a look at it. I have yet to see an instance where the Scantron actually made a mistake. You know, I have many times seen students say, well, look, I chose C on what I wrote on the exam that I took home with me, but I filled in A on the Scantron. Can you give me that point? It's like, no, I got to go with what's on the Scantron. That's sort of what we're doing here. So be more careful next time and make sure you get the right thing. But that seems to be more often than not the reason that there's a discrepancy between what you think you should have gotten and what you got. Is there's a disconnect between what you wrote on your hard copy of the test and what's on the Scantron. And again, if you're not happy with how you did on this first test, the world's not over. We got 2 more tests to go. There's another 25% of the class's grade that's determined by how you do in the lab. There's lots of opportunity to bump your grade up a little bit if you're not happy with where it is. One last thing about the tests. It has been, excuse me, my experience that there is a very high correlation between how students do on the first test and how they do on the second test. Probably wouldn't be too surprising to you. So if you don't like how you did on the first test, you got to buck that trend, and that means probably, more than probably, I think that means something should change. The way you studied, what you did, what you were focusing on, something's going to have to change. Don't go into the second test thinking that everything's going to be better now. That was just a bad day for you. Something should change. If you did well on the first test, take that as positive feedback and don't change stuff, right? You were doing great. Keep doing what you were doing, and you shouldn't have anything much to worry about for the second test. If you don't like how you did on the first test, and you want to have something change, and you don't know what that something could or should be, certainly come talk to me. I'll have ideas for you. I'll ask you a couple questions about what you were doing, where it is that I was confusing to you, and then I'll be able to give you some suggestions about what you might be able to do a little bit differently or better so that you won't have that problem the second time around. The supplemental instructors for the course will also be very good at being able to help you with those sorts of strategies as well. All right. Time to move on. Let's talk now about the content for the course. Actually, before I do, just one other small thing. Please remember that on Thursday of next week, not next week, Thursday of this week, like in 2 days, there's going to be a camera crew in here a little bit. So if you want your mom and dad or your ex-significant other to think you're looking good and you're dressing well, they may be seeing you in the background on some sort of cable TV show, an obscure cable TV show, sometime at the end of this month. So, just a word to the wise, look pretty on Thursday. It turns out Wright State's a public university. We actually can't keep people from coming in to the lecture hall. There's not even any permission forms that they need. So if I wanted to keep the camera crews out, I don't think that I could, but they are coming here to film me and us, so there will be some folks around on Thursday. I'm not anticipating it disrupting the class in any way. All right, so you've been warned. Now, let's get to work talking about biology. honestly, what we've been doing in this class for the first month or so, the first, 9, 10 lectures or so, has been to lay a foundation so that I can talk with you today about this stuff. It's starting to get pretty unequivocally biological. We're getting into stuff that is frankly more interesting than the chemistry, but we need to have that grounding in the chemistry to really appreciate the remarkable things that we'll see happening now in living systems that are manipulating that chemistry. There was fun stuff at the very beginning talking about what it means to be alive and that sort of thing, but there's these rules of chemistry, and maybe the most important thing, just as a big, broad brushstroke to take away from that first part of the course, is there are a bunch of rules that govern how chemistry works, and those rules apply equally to living things and to non-living things. Okay, that's sort of like the big takeaway from that first part of the course. And now, let's see, as the rubber's hitting the road, how it is that living things play a game with those rules in place. You know the rules now. How are you going to win that game? And how could you imagine that living things, in some sense being mindful of those rules, can play the game in a way that lets them pull stuff off that non-living things can't do? That's where we're at. That's what we're going to start talking about right now. We got a word. Not that fancy a word, you've heard it before, but we got a word that describes the sum total of all the chemical reactions that take place inside of an organism. Right? That word is metabolism. Right? It's the sum total of all the chemical reactions that take place. Metabolism. That's what it's all about. And again, the kind of chemical reactions that take place in a living thing are, to a large extent, the same sort of chemical reactions that would take place in a non-living thing, with a big but. And the big but is that we also, as living things, can facilitate certain chemical reactions to take place. The ones that we like, the ones that are suited to our purposes, we can facilitate those, and we can give them a helping hand, make it easier for them to take place, such that they occur more readily and more frequently than the ones that we do not favor. Non-living things just have this sort of baseline background level of chemical reactions taking place. We've got that too, but in living things, superimposed on that background of sort of a baseline set of chemical reactions, there's some stuff that happens because we help it to happen. That's essentially how we're playing a game with these rules of chemistry, is we help some chemical reactions take place as opposed to others. And so metabolism is the sum total. It's all of the chemical reactions that take place inside of a living thing. But most of those chemical reactions, there's that baseline noise that's taking place in all things, living and non-living. Most of those chemical reactions are ones where we're helping them along. In a word, we're catalyzing those chemical reactions. We're facilitating those reactions. And so when you talk about metabolism, you're typically focusing on those chemical reactions that living things are helping along. It's all of the chemical reactions, but it's especially the chemical reactions that we help along. So, in a second, I'm going to show you all of those chemical reactions. Let me tell you one more thing before I shock you with metabolism. And that one extra thing is that a lot of what's going to be in play here is playing with energy. For today's lecture and Thursday's lecture, it's pretty much all going to be about how it is that we move packets of energy around and how we get certain things to happen that couldn't have happened unless we had invested energy into them. That's what that word at the top of the screen is. Thermodynamics is the discipline that studies energy. It's particularly a chemical discipline, that it's chemical energy. But so in the chemistry department, there are people who will tell you they're thermodynamicists. And what that means is that they're schooled in the study of thermodynamics. And thermodynamics is all about the storing and transfer of energy in chemical systems. So a big part of what we're going to be doing for today and Thursday is really talking about thermodynamics. So I've warned you, the next slide may be a little bit scary to you. I'm going to show you a diagram that has the sum total of all of the chemical reactions that take place, the catalyzed chemical reactions that take place in a relatively simple organism that is thoroughly characterized, E. coli, Escherichia coli, a bacteria, a prokaryote that lives in our guts. All right? So don't be freaked out. The next slide shows you all of those chemical reactions. That's it. That's the metabolism of E. coli. Every arrow there, not the pointer arrow, but all the rest of the lines and arrows, corresponds to a chemical reaction that E. coli has an enzyme, a gene that allows it to control and manipulate that reaction. It can help it happen faster. If the circumstances are right, it can actually stop the reaction from happening. Each of those arrows is a chemical reaction. At some level, you look at that and you think, wow, that's a bummer. Do I have to wrap my head around that whole thing? And I think you already know the answer to that. Obviously, I'm not going to expect you to master all of E. coli's metabolism, right? But by the same token, when you see an image like this, take a look at it and appreciate that that is knowable, right? If you really put your mind to it, you could wrap your head around everything that makes E. coli tick. I mean, that's within the realm of possibility. You could look at that. If you spend enough time, you could get it. You could understand the entirety of E. coli's metabolism, right? So it's a mix of spaghetti strands that you can't really see, but it all meshes together. All of those chemical reactions are, to some extent, interrelated and connected with each other. What is it, 7 degrees of Kevin Bacon, right? Any chemical inside of E. coli can be translated or changed into just about any other chemical in 7 or fewer steps. You plop it down on one part of one of these metabolic pathways and you need it to be somewhere else. That molecule needs to change its shape or its structure somehow. You can usually get there in a relatively small number of steps because all of this chemistry is interconnected. And all of this chemistry, again, is knowable. I don't think I've said it plainly. Let me say it very plainly here. Each of those lines as a chemical reaction corresponds to a conversion of one chemical to another chemical. And each of those lines is catalyzed by an enzyme. And enzymes are encoded for by genes within E. coli's chromosome. All right? If you mess up one of those genes, you mutate it, you take it out, you mess up one of those proteins, you know what you've done? You've made a mutant E. coli that is hobbled in some way because it can't do one of the chemical reactions that a normal, wild-type, healthy E. coli can do. So each of those lines corresponds to an enzyme. Each of those lines corresponds to a chemical reaction. Each of those lines corresponds to a gene. If you want to make E. coli a little bit less happy and healthy, interfere with the activity of one of those enzymes. Design a drug that takes that enzyme out of the picture, and you've done something that's going to make E. coli less well suited to carrying out all the rest of the reactions because, again, they're all interconnected. All right, so this is E. coli's metabolism. Again, E. coli, it's a relatively simple, thoroughly characterized, comprehensively understood prokaryotic organism. A chart of human metabolism would be about 100 to maybe 1,000 times more complicated than what you see there. But we're not going to worry about that, right? We're going to focus on something that's manageable and comprehensible, but know that the greater complexity that we have as humans for our metabolism is also knowable. It's just a much harder job to wrap our heads around it, so we'll focus here on the simpler stuff. Do you notice how in the middle here there's a circle? And also, this might be harder to pick out, there's a line of stuff that's happening up here. That line and that circle are central to all metabolism. And that's really what it is we're going to focus on for the next 2 weeks, actually. It'll be not just this week, but all through next week. We're going to focus on those very central aspects of metabolism. We're going to talk about glycolysis, and we're going to talk about the citric acid cycle. Those are the very central pathways that all living things rely upon for playing around with energy. And all the rest, if you look at a picture like this, all the rest seems to just be tacked on to those very central pathways that all living things share in common. Okay? And that's a pretty good indication that what we're talking about here when we do talk about glycolysis or the citric acid cycle is these were metabolic pathways that were established very, very early in the history of life on this planet, and then all the rest sort of gets glommed on to them as life diverges and takes on the ability to do different sorts of things. So one last glance, that's metabolism. That's what's going on inside of an E. coli cell at any given point in time. We're really going to be focusing on what's happening over here. And here is a diagram that just looks at what's going on with energy metabolism within E. coli. So it's actually lifted out of that previous diagram, but now with all the extraneous stuff taken away, the other diagram included pathways, for instance, to help you make all the 20 different amino acids, and pathways to make the nucleic acids, and pathways to make phospholipids, and those sorts of things. Here, we're just looking at those chemical pathways that are involved with energy metabolism, and here is that line. This line is glycolysis, and here's that circle. That circle is the citric acid cycle. All the rest of these molecules are sugars, for instance, that what you do to get energy out of a sugar, if it's not glucose, which is right up here, if it's not glucose, you know what you do? You convert it into glucose and you plug it in through this pathway here. Everything hangs off of glycolysis in the citric acid cycle. And again, these are the chemical reactions that we're really going to focus on for this class. Before we get to much more work about what's going on with these chemical reactions, though, I want to make sure we're all talking the same language. There's some jargon to talk about, and I am just going to be using these words, and I've learned from the previous times I've taught this class that a lot of times I just start talking about them, and students like 3 weeks from now come to me and say, "What's a substrate?" So today, let's make sure everybody knows what all these words mean, because I'll be using them a lot. Here is a chart of metabolic pathways, right? That little circle there corresponds to a substrate. This corresponds to a product. There's a line between them. That's a chemical reaction that's being catalyzed by an enzyme. Let's look. So here's that little circle. Here's the other circle. Here's a couple more circles further down the line. All together, we could call this a pathway. Okay? It's a chemical pathway. It's a path by which a molecule like A can be converted to a molecule like D. All right? There's some steps along the way that you take as you're going down that path. So this starting molecule is often referred to as a substrate. It's just, what we call it. It's a molecule. It's a molecule that's going to undergo a chemical reaction. That's called a substrate. The things that get made from substrates after a chemical reaction has taken place on them we'll call a product, right? So here's an intermediate product. Here's another intermediate product. Here apparently is the big payoff. You wanted to go from A, your substrate, to D. Well, there's definitely the product, but along the way you've got some intermediate products as well that also are substrates, because check it out. B undergoes a chemical reaction as a substrate to become C, a product. All right, but so when you're looking at the left-hand side of one of these pathways, the beginning, we're talking about substrates. And when you're looking at the end result of a pathway, no big surprise, right? That's the product of the pathway. By convention, if somebody draws an arrow between a substrate and a product, they're trying to help you realize that you can convert this substrate to this product. That's what the arrows are there for. And if somebody goes through the trouble of writing down the name of some protein, an enzyme, on top of that arrow, guess what? It's pretty straightforward nomenclature, but I'm your tour guide here today. That name is the name of the enzyme that catalyzes the reaction, that facilitates that reaction taking place. If you took that enzyme away, if you stopped it from doing its thing, you will have effectively stopped the conversion of substrate to product, A to B. That's what catalysis is all about. We'll get more into the nuts and bolts of how catalysis works during the course of today and Thursday's lecture, but that's how these things play out. If you write down the name of a protein, typically, what you're doing is you're saying that's the name of the enzyme that catalyzes this particular reaction. And again, I feel like I'm beating a dead horse here, but you know, this is a pathway. This to convert A to B in this hypothetical world, you apparently need to have a series of 3 enzymes to do that job. There's not one protein that does it going poof, A to D. Instead, you do it in a stepwise fashion. You walk down a path, one by one, a series here of 3 enzymes catalyze the reaction that ultimately delivers to you that product. So again, mostly here we're talking about some nomenclature issues. All right, so there's metabolism for you, right? Big, lots of stuff going on. At finer scale, we're talking about converting substrates to products and catalyzing reactions, typically with enzymes. Fine. But to really see what's going on under the hood and appreciate the magic that is going on with these pathways and that sum total of metabolism, let's pay some attention to the energy that's involved. For prokaryotic organisms and archaebacteria, it is all about managing energy resources. Everything else is secondary. For fancy, complicated organisms like us, some other things do come into consideration, but even for us, we're keeping our eye on the bottom line. It's the energy currency inside of cells is every bit as important as finances are to an institution like Wright State or an individual like you and I with our personal finances. We may sometimes be interested in do other things, but at the heart of it all, you better have your checkbook balanced or you're going to start to find that you got some problems, right? Energy for prokaryotes and archaebacteria is pretty much the only thing that they worry about. The prokaryote that's best at securing and storing and using energy is the winner. It can outcompete any other prokaryote that's less good at securing, storing, and using energy. That's how they win, and that's what they're paying attention to. The rest is all window dressing and luck. But the stuff they can control and really focus on is energy. And I've already told you, there's a study in chemistry, there's a discipline within chemistry where that's what it's all about, is energy, particularly energy in chemical bonds, and that's what thermodynamics is about. So, there are courses in thermodynamics here at Wright State. As a junior, if you, for some crazy reason, pursue a major in chemistry, you will be taking a thermodynamics course or 2. I am a recovering chemistry major, right? I've told you guys, I had a double major in biology and chemistry. I walked that walk. I went through the process of taking thermodynamics classes. Let me give you a little bit, you probably don't care to know, but I'm going to indulge myself and tell you something about my backstory here so that you'll feel sorry for me, right? I knew thermodynamics was going to be tough. That was all the talk on the street, or in the dorms at least, was that thermodynamics is a killer. It's very math heavy, it's hard. So, smart guy, you know what I decided to do? I didn't want thermodynamics to be hard, so I sat in on a thermodynamics class over a summer, just so that when I took it for real, I'd be ready to go, right? Talk about nerdy. That's what I chose to do for the summer. I lifeguarded at the pool at the university, paid some bills that way, and then when I wasn't guarding, I worked the lifeguard schedule around my going to this thermodynamics class. Stupid, but I did it. I wasted a summer sitting through thermodynamics. And then I took the thermodynamics class, and you know what? It was really hard. And there was a lot of math involved with it, but I made it through, moved on. And then I go on to get a PhD, right? And my PhD is formally in biochemistry. A little bit of trivia for you here. You could win a bet someday with this. I am the last person to ever get a PhD in biochemistry from the Pennsylvania State University. The program is still there. It's just I was the last stupid person that could be persuaded to take the courses that you needed to take to become a PhD in biochemistry. There were other options, molecular biology, cell biology, in the same program that I was in. A long story, but I got tricked time and again, and I ended up getting a PhD in biochemistry. All my friends, right? It's like, what classes are you taking this year, Magda? It's like, oh, I'm not taking any classes. What are you taking, Dan? Oh, I'm taking graduate level German and graduate level thermodynamics. It's like, what are you ever going to use that stuff for? It's like, I don't know, but that's what I need to do to get my PhD in biochemistry. So, I know about 10 words in German. And here's the sad part of the story. For the thermodynamics, I had to take a graduate level thermodynamics class. And I started to take the graduate level thermodynamics class, and it was ridiculous, right? I was like, I have no idea what these people are talking about. So you know what they did to help me out? They let me take undergraduate level thermodynamics again, right? So I sat through a summer of thermodynamics. I took the thermodynamics class as an undergraduate. I took a graduate level thermodynamics class. It didn't go that well, so they put me back to undergraduate level thermodynamics, which I took. And then I got to take the graduate level thermodynamics class again. I've spent like 5 years studying thermodynamics, and I never enjoyed even 10 minutes of it, all right? So that's the burden that I bear, and maybe explains some of my issues. But I know a lot about thermodynamics, and I'm happy to tell you today that I can convey to you in the span of about 15 minutes the only bits of thermodynamics that are actually useful and interesting to a biologist, right? 5 years, 15 minutes, right? If somebody could have just sat me down, if I could have made this argument to the people on my PhD thesis committee, I know the 15 minutes, that's all that I need to do, let me out of here, boy, I would have been a happier guy all along. But here's the thing. Thermodynamics, it's the study of energy, right? That's a good thing to sort of have in mind. And all of thermodynamics revolves around two laws, cleverly named laws. There's the first law of thermodynamics and the second law of thermodynamics. There's a third and a fourth, but hardly anybody ever talks about them because all that matters is the first and the second law of thermodynamics. Here's the whole thing in a nutshell. The first law of thermodynamics says that energy is conserved. In 3 words, I've written out for you the entire first law of thermodynamics, conservation of energy. One of those things, what's of? Is that a participle? It's a throwaway kind of word. Conservation of energy is the first law of thermodynamics. I'll say a little bit more about that, but that's it. The second law, guess what? Entropy always increases. You can throw away the always and just go with increases, and you get in 5 words the first 2 laws of thermodynamics. The first law, energy is conserved. The second law, entropy increases. You're done. That's what's going on with thermodynamics. Here's what makes those 2 laws so important and ultimately something that biologists do need to at least be able to tip their hat to. Those 2 things together help us understand what causes some chemical reactions to occur spontaneously and some of them to not. And what enzymes are doing is they're gaming the system to help spontaneous reactions happen faster than they would have happened in the absence of the enzyme. What I said there was actually very carefully chosen words. What enzymes do is they help spontaneous reactions take place faster than they would have taken place in the absence of the enzyme. I did not say that enzymes help chemical reactions take place that would not have happened if the enzyme wasn't there. I said enzymes help reactions that would have taken place anyway take place faster. So there's two kinds of chemical reactions in the world. Those that happen by themselves, and then there are those that don't happen, period. Enzymes only work their magic on that first class. The ones that would have happened anyway, their trick is they help them happen faster than they would have happened. All right? And we'll get, again, I'm getting ahead of myself right now. That's all what spontaneous reactions are about, and we'll talk more about that. But first, let's take a little time to talk about conservation of energy. And there's some fun stuff along the way here. I'll make this as untorturous as is possible. So conservation of energy. Here it is in more words, black and white, just in your face. But stated formally, the first law of thermodynamics says that in a closed system, energy can neither be created nor destroyed. All right? The destroyed part people don't worry about. Usually, it's the created part that gets people excited. Remember a week and a half ago, I told you about how I lost 2 nights of sleep with my perpetual motion machine. The chemistry professor that let me set that up said it's not going to work. And, well, how do you know it's not going to work? Look, on paper it looks great. And I said, it can't work. It violates either the first or the second law of thermodynamics. This is looking like a first law violator to me. All right? Did you know that the U.S. Patent and Trademark Office, going back to Ben Franklin, who set that office up, 200 some years ago, did you know that people have been submitting patent applications for perpetual motion machines back even in Ben Franklin's day? Ben Franklin got so many perpetual motion machine patent applications. Ben Franklin, the guy on the $50 bill, something like that. That Ben Franklin, the key in the jar and the electrocution stuff, that Ben Franklin appreciated these laws of thermodynamics so well that he set up a system that automatically rejects a patent application that claims to set up a perpetual motion machine. It's just automatic rejection. And all that they have to do in the U.S. Patent and Trademark Office is figure out why it is that it's violated either the first or the second law of thermodynamics. And they have a stamp that says violation of first law. Or I have another stamp that says violation of second law. If they look at your patent application and it looks like you're creating energy where energy wasn't before, they trot out violation of the first law stamp. You can't create energy where energy was not before. You can't get something moving perpetually. You can't extract energy from a system perpetually. There's a finite amount of energy in any closed system. All right? Now, you can play games with how you define the boundaries of the system. You could say the system is what's going on inside of a cell. Okay, that's one closed system. You could say it's what's going on inside of an organism, an ecosystem, a planet, a solar system. But within a certain boundary, depending on what you're talking about, you can't just pull energy out of nowhere. And it turns out that one of Einstein's most important insights, you've all heard E equals mc squared, right? That ultimately is rooted in the first law of thermodynamics. E, energy, right, is equal to mass times the speed of light squared. So what Einstein figured out with his, phenomenal insight was that, in fact, the first law of thermodynamics is you can convert energy to matter and matter to energy, but that's it. There's a finite amount of energy or matter in any system that you might have, and you can swap them back and forth between each other, perhaps. It's hard to do, but it can be done. But that equation defines the sum total of the energy in a system. And again, the first law of thermodynamics says you can't be getting energy where there wasn't energy before, right? Finite amount of energy. And by the same token, you can't do away with that energy or matter. It's there, it's permanent, it never changes. So that's what's up with the conservation of energy. Let's talk about the second law of thermodynamics, and that is that entropy increases. And entropy is fancy talk for disorder. I don't know why they stick with entropy. Disorder does every bit as good of, it's what normal people would say. It's every bit as good a descriptor as entropy to describe what's going on here. But in any system, there is a natural tendency for disorder or for entropy to increase. So here are two real-world examples, right? If you start off with a system like this beaker, I think, right, where there's order, right? There's a bunch of yellowish molecules at the bottom of the beaker and a bunch of bluish molecules at the top of the beaker. Are you going to be surprised if over time that the yellow balls and the bluish balls just sort of jumble together? Dye molecules, water molecules. That's just the way nature behaves. You're increasing the disorder. There had been order on the left where you had a higher concentration of blue in one part of the container, one part of the system, relative to yellow. And over time, because disorder increases, because entropy increases, you expect this sort of diffusion to take place. This is a molecular or a marbly kind of example of how disorder plays out. Check out this guy here. Which of these two scenarios is more plausible? This guy has some energy associated with him, and he's got bricks, and he's throwing them randomly to his left and his right. On the left, the random throwing of the bricks results in a nice, neatly ordered brick wall. To his right, I can't even tell which way the guy's facing. But on the one side, he's making a wall. On the other side, he's making a jumble of bricks. Which of those two is more likely to happen in the real world? Do you think bricklayers do that? They close their eyes, they get their bricks, and they just throw them? No, of course not. Because the brick wall, you're seeing order, right? There's something that's there that's organized. On the right, there's just a jumble of bricks. That's because naturally occurring systems, in natural systems, disorder, entropy increases. You don't expect spontaneously for things to appear to be more ordered as opposed to less ordered. So every time you transfer energy, this guy's transferring energy to his bricks, right? Every time you transfer energy, it results in an increase in disorder. Now, let's pause and take a biological look at this. That very first time we met, you thought I'd never even remember what we talked about the first time, and yet I keep touching back to that first lecture. One of the things that we kind of agreed, that I at least foisted upon you, and you were kind enough and polite enough to not argue with me about, but I suggested that one of the things that lets us distinguish between living things and non-living things is that living things are more ordered than their surroundings, right? That was one of the four things, the four criteria for living things. And I've just told you the second law of thermodynamics says that entropy must increase, disorder must increase. How can you reconcile those two ideas at the same time? How can living things be more ordered than their surroundings, and yet this inviolable, you can't break this law of thermodynamics. The second law says entropy increases. Every time you transfer energy, you set up a system that's less ordered than it was before. Here's how living things pull this off, how they manage to be more organized than their surroundings. They pay an entropy tax. Nature is absolutely insistent that disorder in the system must increase. But what living things do is they play the game, right? They negotiate with nature. And they say, hey, here's the thing, Mother Nature. I'd really like there to be a higher concentration of whatever you like, sugar, in this little part of the world, inside of myself, than outside of the world. I want this to be an ordered system. And I know you don't like that. So here's what I'd like to do to make it worth your while. Mother Nature, you let me have a little bit of order over here, and in exchange, I'm negotiating this with you. In exchange for this nice little ordered spot that I like, I'm going to make the rest of the world, the rest of this system, more disordered still. Right? So there's a certain amount of disorder in the system to begin with. I'm going to make the rest of the system even more disordered than it was before, in exchange for the ability to make something in a little tiny spot more ordered than it would have been otherwise. So there's a negotiation, there's a brokering that takes place. To set up something that's ordered, you need to pay a price of increased disorder somewhere else. To set up order inside of a cell, what ends up happening is you increase the disorder outside of the cell, and you increase it disproportionately. It's not just you got 1 unit of order inside and you make 1 unit of disorder outside. You actually do 1 unit of order inside in exchange for maybe 5 or 10 units of disorder on the outside. Okay? So that's how it is that living things can pull off the trick of being more ordered than their surroundings. And let's also talk here a little bit about this conservation of energy deal. You guys are all college students, so I suspect that you'll understand this immediately. But I've had neighbors in the past who needed to be taken to school on this idea. It turns out that a car left with its engine running will eventually stop, even if you're not driving the car. Right? And I had a neighbor once who said, I don't know what's wrong with my car. It's just, I was warming it up for like 4 hours and now it won't work. I wanted it to be nice and warm. And it's like, maybe you ran out of gas. No, I couldn't have run out of gas. I wasn't driving it. I was just running the engine. Yep, that's what was going on. Right? There's a finite amount of energy stored in that gas tank. And even if you're not moving, right, that energy is getting burned up, it's being consumed as you're idling your car. What happens to the energy that had been stored in the covalent bonds of the hydrocarbons in the gas tank? It wasn't used to move the car. It was used actually to generate heat. Maybe not intentionally to generate heat, but warming up a car implies that that was the guy's intention, right? But energy, it's consistent now with the first law and the second law. If you're moving the energy from the covalent bonds in the hydrocarbons into carbon dioxide and water molecules, there's going to be that transfer of energy, even if it's not used for motion, will result in a generation of heat. And heat then turns out to be a pretty good measure of disorder. The faster molecules are moving around, generally the less ordered they are. So there's a direct conversion between heat and disorder. Okay? But let's say that instead of just idling your car, you want to get your car to move from point A to point B. You're going to do that by burning hydrocarbons, taking these high-energy, nonpolar covalent bonds, breaking them up, releasing that energy, creating low-energy molecules like carbon dioxide and water. Low energy, high energy, you're releasing the energy, you're moving that energy from one thing to another. You want to use that energy to cause your car to move, right? That's what you want to do. How efficient is that process? Have you ever stopped to think about how much energy is actually stored in gasoline and how much of that energy gets used to translate into motion of your car as opposed to having been lost as heat? Because you probably realize that when you're driving your car, the engine gets warm. That's entropy. You're transferring energy from some covalent bonds into something else. That energy transfer, the second law of thermodynamics, that transfer must result in an increase of entropy. It must result in something getting heated up. Do you know how much heat gets lost by an internal combustion engine? It blew me away the first time that somebody sat me down and explained to me the actual efficiency of an internal combustion engine. 75% of the energy stored in the bonds of gasoline gets lost as heat. Only 25% of that energy is actually used to make your car go from one place to another. At optimum efficiency, that's the best that about 100 years of clever people in Detroit, Michigan, thinking about how to tweak and improve car engines, the best that those engineers have come up with after 100 years is a system that's only about 25% efficient. Kind of disappointing, don't you think? If you could make a car engine that was 50% efficient, you could double the gas mileage. You'd use whatever, half the gas that you had been using before. If you could make it 75% efficient, it seems like there's a lot of room for improvement. Right? So how ashamed should we be about American engineers who have been working on these engines for 100 years? Is this an embarrassing thing? We know from the second law of thermodynamics that it cannot be 100% efficient. The second law of thermodynamics says you must increase entropy, at least a little bit, but you must increase entropy every time you're trying to do those types of for each energy transfer, right? So it can't be 100% efficient, but seriously, can't we do better than 25%? A lot of room for improvement. Well, this might help put that into some perspective for you. Maybe 25% efficiency isn't that bad, because living things over the course of about 3 billion years of negotiating with nature for energy transfer of energy from, say, glucose to ATP molecules, at peak efficiency, those energy transfers happen at 34% efficiency. Glucose has 684 kilocalories of energy per mole, and you capture only 34% of that when you're using glucose to make ATP molecules. All the rest, 2/3, lost as heat. Every time you crack one of those glucose molecules because you want to make some fresh ATP, you're throwing away 2/3 of the energy that had been stored in that original glucose molecules. So, car designers, at 25%, give them another, 3 billion years, and maybe they can get up around 34%. But at 25%, they're actually not that bad. That's probably about as good as you could expect human engineers to be able to design a system to come up with. But take a step back and think about the implications here. Every time you transfer energy, you pay a hefty entropy tax. That's one of the best arguments that I've ever heard for being a vegetarian, right? You could feed many more people off of a given unit of land as vegetarians than you could if you were omnivores eating plants and animals. And what's going on is pretty simply this. You've got plants that are harvesting energy from sunlight, there's a transfer of energy that's involved there. So there's some energy that's being lost, but we don't know of things that are particularly good at sucking energy straight off of the sun. So let's not worry about that one. We're stuck with that, I suppose. But you got plants that capture that light energy and make a sugar out of it. And then you got some animal that comes along and eats the plant and breaks up those sugars to be able to make their own sugars and their own ATPs. There's a 2/3, 66% energy entropy tax that happens during that transfer. And then you come along and you eat the animal, and 2/3 of the remaining energy gets lost for you to be able to get those energy-rich molecules and to do something with them. And if you put another animal or something else in between, right? You put you got your plant, you got your cow, and then you got a bear, and then you eat the bear, it's all the worse still, right? The higher up the food chain you are, the worse you're doing in terms of energy transfers. Now, again, this is a very logical, very compelling argument. I like chicken and some other things, so I haven't gone the whole way, but I understand the logic, and from a thermodynamics perspective, being vegetarian actually makes an enormous amount of sense, and it's because of that energy tax. Last week, I was telling you, what if I wanted to get a Pepsi out of a vending machine, right? A dollar bill is going to come in really handy for that, right? A $100 bill might make you feel pretty good about yourself, but it's not very practical. Glucose is that $100 bill. There's a lot of energy, 686 kilocalories per mole, big chunk of change by energy standards. But to get stuff done inside of a cell, what you really need are those dollar bills, the ATP molecules. I've just told you that when you take your glucose to the bank, you're getting only 34% of the ATPs that the bank could have given to you based on the energy that was stored in the molecule. Think about how outrageous an exchange rate that is. If you took a $100 bill to a bank and they gave you $34 back, not too happy. As living things, we smile and we say to Mother Nature, thank you very much. We appreciate having done business with you. I'm looking forward to seeing you again. Because that's the best deal that we have, as living things, have been able to negotiate after 3 billion years. Right? If you went to Right Pat Credit Union every day with a $100 bill, how long would it take for you to say, thank you for giving me the $34, right? Wouldn't happen the first day. You'd probably be pretty mad. Wouldn't happen the second day. By what, the 50th day? That's just the way it goes. I guess that's the best deal. I've looked at all the other banks. Nobody else will give me a better deal. Thank you very much for taking only 2/3 of the energy and squandering it and shredding it up and throwing it into the air as heat. That's what's going on. And we like it, oddly enough. So that's what thermodynamics is all about. I've been telling you now some things about cellular energy and glucose and ATPs. I'm going to backfill with some pictures here now for what's going on over here. You know, every time I show this picture, I find myself wondering, what's the point of this picture? Is it really necessary to say? But maybe it is, so I'm on the fence. Here's all that's going on. Check it out. You know, somebody on top of a diving board has what we call potential energy, right? And somebody that's jumped off the diving board and is in the water, guess what? They have less potential energy. They've expended their potential energy. It's gone, right? And the energy of motion is kinetic energy. So what we're really talking about here is somebody on top of the board has the potential to have kinetic energy, this energy of motion. All right? This is starting to look disturbingly to me like thermodynamics. It's just the tip of the iceberg. Believe me, the math gets a lot nastier the further in you go. But I think you can appreciate, based on all I've been saying now this morning, that energy is a big deal. Living things pay a lot of attention to the energy associated with systems and chemical bonds. And thermodynamicists have given us a useful term, a concept, that describes the total amount of energy associated with a system. And again, you can define the system however you like. Draw the lines wherever you like. Sometimes you say the system is this cell. Sometimes you say the system is a subcomponent of a cell, a membrane-bound compartment like a mitochondria, right? So you draw the lines wherever it's necessary. But within a system, there is a total amount of energy. And thermodynamics folks have said, hey, we have to have a word that describes that total amount of energy. And guess what the name of the guy was that really pushed this idea? His name was Gibbs, right? And so Gibbs, for the longest time, was telling all his thermodynamics buddies, they'd get together at meetings, they'd drink some beers, they'd smoke some cigars, and he'd say to them every night, hey, look, we got to have a nice single word that describes the total amount of energy in a system, all right? We got to have a word. What are we going to call that word? It's the Gibbs free energy of a system, usually just shown as G for Gibbs free energy. All right? So maybe there's a lesson to be learned there. You want to be famous, go out drinking with people and smoking cigars with them, tell them you need an idea or a name for a simple idea, and they'll name it after you. So if a system's got a lot of energy, you know what we say? We say it has a lot of Gibbs free energy. We say it has a high G, right? Things that have a lot of energy, like, for instance, a stretched out rubber band, right? Or a gas tank full of gas, things that have a lot of energy associated with that system are typically less stable than things that aren't stretched out. Which way do things go? When you have a stretched out rubber band, and you let go of it, is it going to get more stretched out, or is it going to become less stretched out? If you have a less stretched out rubber band, is it going to stretch out on its own? No. Stretched out rubber bands aren't very stable. Not stretched rubber bands, very stable. They'll stay not stretched for a really long time, right? And so high energy systems, things with lots of Gibbs free energy tend to be less stable, but there's also a capacity at least to do work. For a chemical reaction to be spontaneous, and let's not be intimidated by the word spontaneous. In this context, it means the same thing as it does in everyday English. It means that it's going to happen by itself, right? Just poof, it happened, right? Things that happen spontaneously are things that go from high energy in the system to lower energy in the system. That is the definition of a spontaneous chemical reaction, going from high free energy to low free energy, or to lower free energy. And as a result, what you'll say is that delta here, this is the uppercase Greek letter delta, delta G, the change in Gibbs free energy, if it goes from a high value to a lower value, we say that that's a negative delta G. What you're actually doing is you're subtracting the end energy from the starting energy. If that's a negative number, you've got a spontaneous chemical reaction. Okay? And so, what is the total amount of energy for a system? Gibbs free energy. It's the total amount of energy. It turns out that the Gibbs free energy is a function of really just two things. It's H minus Ts. H is enthalpy. It's the energy stored in covalent bonds. S is entropy, the disorder of the system, and T is the temperature. So temperature times disorder, they're related ideas, they're factored together. G equals H minus Ts. Okay, that's the total amount of energy. There's only two balls you need to keep track of when you're thinking about energy in a system. It's the energy stored in covalent bonds, and it's the energy stored in the order and temperature of the system. Okay? But if you go from high energy to low energy, we say you have a negative delta G. A negative delta G means it's a spontaneous chemical reaction. going from the top of the diving board to the bottom of the diving board, spontaneous. Going from a little cluster of molecules huddled together to a diffuse spread, more disorder, right? High energy, low energy, spontaneous reaction. Taking a cluster of atoms that are linked together by covalent bonds and breaking those bonds goes from high order to low order, high enthalpy to low enthalpy. Bottom line, spontaneous reaction. You go from order to disorder, high energy to low energy. All three of these things are spontaneous. This one is spontaneous largely because of the increase in entropy. This one is spontaneous largely because of the decrease in enthalpy, the energy stored in the covalent bonds, but it's also having an effect on the disorder as well. More important than anything else, all three of these would be characterized as having a negative delta G. We've gone from high order to less order as we've gone from the top of the screen to the bottom of the screen. And so you can juxtapose this story right next to those, and it's all there as well. All right, so that's what's up with a spontaneous reaction. And let me say a few more things to you now about what's going on with cellular energy. You know, I really can't emphasize it enough. For a prokaryotic organism and for an archaebacteria, this is all that matters. It's all that they focus on. Everything that a prokaryote does in one way or another is tied to more efficiently capturing or using or transferring energy. Okay, so let's talk about, from a cellular perspective, where the energy's involved and how it is that we can get spontaneous reactions to occur, and more importantly, perhaps, how you get non-spontaneous reactions to occur. So what's on the screen now is simply an example of a spontaneous chemical reaction. There's nothing a cell needs to do to help this reaction take place, right? It's going to happen on its own because you started out with substrates, here reactants, they're synonymous. You started out with substrates or reactants that have a lot of energy, and you end up with products that have less energy, right? That is the definition of a spontaneous reaction. All you have to do is realize that the delta G is a negative value. The amount of energy here minus the energy here is a negative value. Delta G is less than 0, right? That is the definition of a spontaneous reaction. You go from here to here, and along the way, as the reaction progresses, you release a bunch of energy, okay? Nature loves this. It'll allow it to happen pretty much whenever you like, because what you've done is you've increased the disorder of the system. Nature's all about scratching its second law of thermodynamics itch, okay? Loves it. You might wonder, why do molecules like glucose hang around at all in the first place, right? This is an energy-rich molecule. Why not just poof, have it all change, spontaneous combustion kind of things? You guys hear those stories? You know, people sometimes just poof, spontaneously combust. Maybe what's going on is all their glucose molecules released all their energy in one big poof, and their whole body gets consumed and off they go, right? Why doesn't that just happen? Why can we hold glucose in our hands without watching it catch fire? Why do our hands not catch fire even when we're not holding glucose? Because of all the energy stored in the molecules in our hands. Well, our bodies will burn, and glucose will burn. It's just that they're in a pretty stable conformation right now. To get to the point where you can release that energy, you have to jack the system up a little bit more to get to what's called an activation energy. We'll talk more about that in a bit. But bottom line for where we're at this point in the discussion, going from high energy to low energy, falling off a log easy. And in fact, that's an example of a spontaneous reaction, falling off a log, all right? And that's in contrast to a not so spontaneous reaction, an endergonic reaction, a reaction that requires the input of energy in order to convert substrates to products. How do you ever make that glucose in the first place is also a pretty good question, because here we could be talking about things like carbon dioxide and water. How do you manage to get those things together so that you end up with glucose that has a lot of energy, so that later on you can have the spontaneous reaction of releasing that energy? Right? Well, the short answer is you have to invest energy to be able to get a reaction to take place that has a positive delta G. To be able to go from low energy reactants or substrates to high energy products, you're bucking the trend and you have to invest energy. You're increasing the order of that system, at least at that level. So how do you pull that off? Short answer is this. You can get non-spontaneous reactions to occur by coupling them to even more spontaneous reactions. That's the game that living things are playing. When we need to go against this tendency toward increased disorder, when we need to have a positive change in the free energy of a system, you pull it off by linking that chemical reaction to one that is even more favorable from an energetics perspective. You have to tie the two of them together. You essentially say to nature, here's the deal. I want to have a glucose molecule, and in exchange for letting me have a glucose molecule, I am going to link the creation of the glucose molecule directly to a bunch of things that release even more energy as disorder. I'm going to burn more molecules up with even more energy than it takes to make a glucose, just so I can make a glucose. It's that inefficient transfer of energy again, and we facilitate that sort of thing. So again, what you can do is you could hook this spontaneous chemical reaction up to this not spontaneous chemical reaction, and the two together result in a favorable delta G. All right? The bottom reaction by itself, unfavorable delta G. Top reaction by itself, very favorable delta G. You put the two together, and you could find yourself in a circumstance where the total is a favorable delta G. Okay? It's bookkeeping. In nature, pays close attention to how you're keeping those books. All right, well, we're at the point now where I'm going to start talking with you about how it is that we use enzymes to facilitate all of this, to be the intermediary with nature that links reactions together and helps some chemical reactions take place faster than they would have otherwise. That's a really good breaking point. And so let's call it a day. Come back on Thursday. Look sharp, right? And we'll start talking about enzyme kinetics and finish off this outline then. All right, see you guys Thursday.